Hello everybody. In today's lecture, we shall study about the internal structure or the transverse section of dicot and monocot root and stems. In this slide that you can see the ground plan of the stem and root of various plants. The ground plan reveals the arrangement of the vascular bundles which vary from dicot and monocot stem and roots. The transverse section of a typical dicot root shows the following characters. The outermost layer is the epiblemma. In case of root, instead of epidermis, we use the word epiblemma, which is made up of parenchymatous cells. Single layer, stomata is absent, cuticle is absent, and the epiblemma gives rise to the root hairs. Just below the epiblemma lies a large layer of cortex made up of loosely arranged oval or round-shaped parenchyma cells. These cells store reserved food materials. Below the cortex is found the endodermis, which is made up of barrel-shaped cells. The inner and the radial walls of the endodermis are thickened by suberin, and therefore these are otherwise called as Casparian thickenings or Casparian strips. The stele is the central part of the root, which is lined by the pericycle, which is single layered. The root hairs usually originate, or the lateral roots originate from the pericycle. The vascular bundles are radial in arrangement, the xylem and phloem alternating each other. In a typical dicot root that you see, for example, in Sisa, there are 4 xylems and 4 phloems which alternate each other. This is a tetrarch arrangement of the xylem, and it is exarch in nature, since the metaxylem cells are present towards the center, which are polygonal in shape, while the protoxylem cells face the periphery. Pith is absent. Let us now look at the internal structure of a typical monocot root. Some examples of monocot root are the canna root, banana root. All these are examples of monocot. The internal structure of the monocot root shows the presence of the outermost layer, just like what we saw in case of the dicot root, that is the epiblemma, which is made up of single layer of cells which are compactly arranged and which gives rise to the root hairs. Below the epiblemma lies the parenchymatous multilayered cortex. The innermost layer of the cortex is the endodermis, made up of barrel-shaped cells, and also shows the presence of thickenings or Casparian strips. The stela region is the central part of the root, lined by the pericycle, which is single-layered, made up of parenchyma cells. The vascular bundles are radially arranged and alternating. The xylem and the phloem alternate each other. The xylem shows exarch condition. The phloem comprises of the companion cells, seed tubes, parenchyma cells. The pith is the centralmost region of the stele, which is made up of parenchyma cells. Let us now study the transverse section of a dicot stem. The picture that you see here is that of the sunflower or a helianthus stem, which is a little more complex compared to that of the root that we studied earlier. The TS of the dicot stem shows the presence of the outermost epidermis, which is single layered, made up of barrel-shaped cells, lined by cuticle. The cells are arranged very compactly without any intercellular spaces. Since it is a dicot stem, there can be small multicellular hairs that may arise depending upon the type of plant. Cortex lies just below the epidermis, which is multi-layered, differentiated into outer hypodermis, followed by a few layers of collenchyma, middle chlorenchyma, and lower parenchyma cells. Just below the cortex is found the endodermis, which is single-layered. The cells of the endodermis are barrel-shaped, compactly arranged, and contains abundant starch grains. And therefore, the endodermis is also called as the starchy sheath. The central part of the stem shows the presence of the stela region, surrounded by the pericycle, which is made up of compactly arranged parenchyma cells. The vascular bundles are arranged in a concentric ring-like structure. Each vascular bundle is conjoined, collateral, and open. There may be many bundles of vascular bundles in the stellar region. The xylem is endark, with protoxylem facing towards the pith and metaxylem towards the periphery. Between the xylem and phloem is seen the presence of cambium, which is the intravesicular cambium. The pith lies in the center, made up of compactly arranged parenchyma cells. In between the vascular bundles are seen the medullary rays. In this slide, we can typically see the internal details of a monocot stem. The best example is that of a grass stem or a maize stem, where the outermost layer is the epidermis surrounded by a thin layer of cuticle. Below the epidermis is a large layer of multicellular region or the ground tissue, which may be differentiated into sclerenchymatous cells, chlorenchyma cells, and parenchyma cells. The vascular bundles are numerous and are scattered in the ground tissue. The bundles are collateral, endark, and closed. Cambium is absent. The metaxylem vessels usually are large, while the protoxylem cells are small. The protoxylem cells may show the presence of a small cavity in the center. Outer to the xylem are seen the phloem. The entire vascular bundle is surrounded by a sclerenchymatous tissue which forms the bundle sheath. The monocot stem does not show the presence of the pith. Thank you for watching.